Okay, hey guys, welcome back to the channel. So today I'll cover chapter 5 for biology, which is mitosis, and we'll talk about its significance and why it's necessary as well. So some terms in this chapter will be used interchangeably with chapter 6, which is about translation and transcription. So I'll be uploading that one after this. So if you get confused about some terms which I'm using, I recommend to watch that video as well. Okay, so let's just start. It's a very short chapter and it isn't too challenging, so we could clear this really fast. Okay, so the first thing we need to know is about the important terms in this chapter. So firstly, it's about haploid and diploid cells. So haploid is about haploid and diploid talk about the number of chromosomes. Um, then it like the number of chromosomes in the cell. So haploid contain N, which is one chromosome per pair, while diploid will contain two sets of chromosomes for every pair. So every pair here means the 23 chromosomes that we have. And why is mitosis even necessary in our body? So first of all, it allows asexual reproduction because when the cells which are produced, they're producing two genetically identical daughter cells and they're haploid as well. And it's a nuclear division. And it can help to it plays a role in tissue repair. So take a note in the exams, they will usually ask under tissue repair about mitosis, um, repairing cells. It doesn't repair cells, it replaces the damaged cells, so it repairs tissues. It allows growth in multicellular organisms and also in the immune system, which we learn in chapter 11. We'll be talking about how cloning of B and T cells by mitosis results in an immune response. So the next thing we talk about is the structure of DNA before we talk about how DNA is separated during mitosis. So the DNA molecule, and we'll talk about the structure of the DNA molecule itself in the next chapter, but just consider this as a DNA molecule. So DNA molecule will wrap around something called a histone protein, and the reason why it wraps around is to make the structure of DNA compact. And when it wraps around histones, it forms a nucleosome. So if you can look over here in the bottom right image, so when the DNA molecule will wrap around the histone protein, it'll form a nucleosome. And in this segment of DNA, which is between the nucleosomes, you can call that linker DNA, which connects the two nucleosomes. So once the DNA coils around histones, there'll be coiling and supercoiling. So you don't really need to know about the supercoiling and coiling. So you just need to know that it coils and it forms the structure that we know as chromatin thread structure, which is present in the nucleus. And chromatin can exist in two forms, which is euchromatin and heterochromatin. So euchromatin is basically the loosely coiled version of chromatin thread, and it contains active genes. And this is the version we talk about mostly during interphase, where the DNA replication happens. And heterochromatin is the tightly coiled genes which are inactive. So this happens once the chromatin thread will coil and condense to form chromosomes. So chromatin thread which will coil and condense and will form a chromosome, and a chromosome will contain one DNA molecule. So during interphase or mitosis, or sorry, interphase will produce during interphase there'll be DNA replication, correct? So it'll produce a chromosome which has two sister chromatids, and each chromatid will then have the DNA molecule. When each chromatid is one DNA molecule. So we have to talk about the structure of a chromosome more in depth. So in this, we'll talk about the kinetochore, the centromere, and the telomere. So let's talk about the kinetochore first. So the kinetochore is its outside region, out of which is attached to the centromere, and it's also bound to the microtubules. So it binds to the centromere and the microtubules. So the function of the kinetochore is that it's involved in the separation of the centromere during anaphase, and it can also sense the tension of the microtubules and see if they're positioned properly or whether they're attached properly as well. And then once they sense this, they can convey a message to the cell to indicate that cell can continue with anaphase. So the cell does have checks along the way of mitosis to see whether it can proceed to the next stage of mitosis. And this is formed during S phase, and there are special proteins which can bind to the centromere. So this is taken from the biology textbook. So the next thing you talk about is the centromere. So the centromere itself is a region of specialized DNA which forms a link between the chromatids, and it would In this case, the only function it does that we need to know about is that it provides a foundation for proteins to assemble and so that the kinetochore can form. And this is important because if the kinetochore cannot form, the centromeres won't split and the chromatids won't separate during anaphase. The next part is about telomeres. So telomeres are these end regions of chromosomes. They are on each end or the ends of the chromatids, and they're regions of repetitive nucleotide sequence. So on one end there'll be a lot of cytosine, and the other will be guanine. So we'll talk about complementary base pairing more in depth in the next chapter. So that's all you need to know, that on one side there's rich cytosine and the other side is rich in guanine. And there's an enzyme called telomerase which will add which will add more repeated bases at the ends of telomere after the cell division. So this prolongs cell life because without telomeres, the essential DNA will be lost. So the issue with DNA replication is that the ends of molecule may not be included in the replication because the enzyme will not run all the way to the end of the DNA. So it will stop a bit ahead. So if you extend the entire chromatid length by adding DNA that does not code for anything, it will it will go a bit more in front, which will cover the vital DNA and also some useless bases. So what's the function of the telomere? So it protects the genome from degradation and preserves the vital DNA. Otherwise, it will be lost because the DNA will keep shortening, and then eventually the vital genes will be lost. And this will prevent the cell death, of course, and it allows cell to divide continuously because without telomeres, the cell division will eventually slow down and it will stop. So the cell cycle is comprised of multiple stages. So one of them is the interphase, which is comprised of three stages itself, which is G1, S, and G2. And we'll talk about mitosis, which is prophase, metaphase, anaphase, and then telophase. And then the last stage is cytokinesis. So you need to take a note that telophase and cytokinesis is in fact two different stages. So what happens during the interphase? So in G1, which is the largest phase of the cell, so this is where the cell carries out its normal function. So this is where it'll do protein synthesis. And in S phase, this is where the cell starts preparing for mitosis. So this is where the DNA will replicate, and now the chromosomes will have two identical sister chromatids, and then kinetochores are also made. And in G2, this is where the cell will continue to prepare for mitosis, and the most important part about mitosis is producing the microtubules for the centromeres to separate and drag the chromatids towards the opposite end. So it will have intensive protein synthesis at gap 2, and it'll produce tubulin, and tubulin is what's used to make microtubules. And this is also the stage where centrioles will replicate because you need to have a centriole at each pole. So the first stage of mitosis is prophase, and we usually divide prophase into early and late. So in early prophase, the nuclear envelope and the nucleolus will start to disappear. And at this part, the nucleolus and the nuclear envelope will somewhat look intact. The chromatin thread will coil and condense, and then it will form this darkly stained, shorter and thicker appearing chromosomes. So the chromosomes will be visible at this stage, and the chromosomes will have kinetochores attached, and then there will also be two sister chromatids. So if they ask you to describe the structure of chromatin thread or DNA at prophase, so this is what you will write. And the centrosomes or centrioles will start to move towards opposite poles. So in the late prophase, the nuclear envelope and nucleolus will have disappeared, and the centrosomes have almost reached the opposite poles, and the mitotic spindle will begin to form once they have reached to the opposite poles. Okay, so let's look at this picture down here. So the nuclear envelope will disappear, which means it will break up into small vesicles, and then the nucleolus will also disappear. And the chromosomes, as we discussed, they are seen as two identical chromatids. And then one chromatid is one DNA molecule, as I mentioned, and centrosomes are moving to opposite poles. And once they reach the opposite poles, they start forming the spindle. Okay, so metaphase is where the DNA chromosomes will lie along the equator of the cell, and the centrosomes would have already reached the poles, and then they will begin to act as MTOCs, which is organizing microtubules to form the spindle fiber. They will attach to the kinetochore at both sides of the centromere. So this is preparing for the chromosome and the centromere to split. Okay, so the anaphase is where the chromatids will split. So microtubules are attached to the kinetochore, and then they will shorten from the kinetochore end and also from the centrosome end, which will create this pulling effect, which will cause the centromere to split, and chromatids will be pulled to the poles. And you need to take note that the way the chromatids split, it will be pulling the centromere first, and then the chromatids will be following it. So the centromere is pulled, and then the chromatids are behind it. So if they ask you to draw how this splits, you have to take note of this. So the telophase is the last stage of mitosis, and in this stage, the chromatids have already reached the opposite poles after they've been pulled to them. They will uncoil, and then they will form this loosely coiled structure of chromatin thread or euchromatin, and then furrows will have been formed on the cell surface membrane. And we'll talk about cytokinesis, and cytokinesis is not a part of telophase, but it will follow telophase. So in cytokinesis, the cell surface membrane, it will constrict from both sides, and then the furrow will deepen until the cells are cleaved, and it will form two separate daughter cells. And obviously, now that you have two sets of chromatids, you'll need to form two nucleoli and two nuclear envelopes. And the spindle fiber, which was used to drag them to opposite poles, they will be broken down in the cytoplasm. And in plants, this is different because they occur without centrosomes. That's the first key feature. And then for forming new cells, the Golgi body will come and place down new material. And then this material will mature to form a cell wall. So this material is involved in cell wall synthesis. Okay, so let's look at this picture here. So the nucleolus is reforming. Nuclear envelope is also reforming. Centrosomes are at opposite poles, so one will go to each. Spindle fibers are being broken down here, and cytokinesis will follow this. This is where the cytoplasm will constrict from both sides until the furrows deepen, and then it'll be cleaved. And then, so this also says that the chromatids, which have reached the opposite pole, they will uncoil. All right, so that's the end of mitosis. And then we also have to talk about cancer and stem cells. So what exactly is cancer? Okay, so cancer is uncontrolled cell division by mitosis. It results from spontaneous change in gene structure. This is known as a mutation, and this results in this cancer-causing gene, which we call an oncogene. And this is usually caused by mutagens or carcinogens. Carcinogens are mutagens which result in formation of oncogenes, and this can be radiation like UV light, tar in cigarette, mustard gas, and viruses. So you have to also take note, I have to ask this before in exams, that viruses can act as mutagens and carcinogens. So cancer cells will not be removed from the immune system, and then tumor can get bigger. If once it gets supplied with blood and lymph vessels will start to form, it gets recognized as a tissue. And then tumor cells itself can enter the blood and form other types of tumors in other places. So this is known as metastasis. If the tumor is malignant and not benign, which allows spread of cancer from point of origin to other parts via blood or lymphatics to form a secondary tumor, which is at another location. And what are the features of cancer cells which allow cancer to take place, or sorry, not allow cancer to take place, for tumors to form? So there's no programmed cell death, meaning they will continue to replicate and replicate. They do not die off by themselves. There's rapid DNA synthesis and uncontrolled protein synthesis. This is important because they're going under cell division rapidly, and they have shorter interphase, meaning they skip majority of they don't really have a function, they just skip interphase. You could say they have a shorter interphase because they just go to the cell division part, and then they'll form this undifferentiated, meaning they have no function, unspecialized mass of cells, and they can form tissues, and they have no function at the site of origin. And we can also talk about stem cells because they are somewhat similar to cancer cells because they can also divide unlimitedly by mitosis, and they can produce new stem cells, or they can produce differentiated cells, which means the cells which are stem cells, which they can differentiate to form a different type of cell. For say, example, red blood cell. So if it goes from stem cell to red blood cell, it will lose its nucleus. It'll be able to make hemoglobin and stuff like that. So it'll obtain the features to become that type of cell. And it can differ into a variety of cells, and it'll depend on its potency. So totipotent, meaning it can differentiate to any type of cell, and multipotent is only a few types of cells. So for example, bone marrow, it can only become blood cells, monocytes, neutrophils, and lymphocytes. And what exactly would be stem cell therapy? So stem cell therapy is when you add stem cells into the body. And what exactly do we use stem cell therapy for? So this allows repair of diseased, injured, or dysfunctional tissue. So you add stem cells, and then they will differentiate into that type of cell and replace the damaged or diseased cells over there. You can also test for drugs for the effectiveness and side effects, because you can allow stem cells to grow and mature into tissues and organs. And you can also investigate how diseases can affect the body. Similarly, okay, so that's the entire chapter for mitosis. It's a very short chapter, so we'll do a few questions to test your understanding. So question 17, so this is from one of the past papers. At which stages of mitosis are chromosomes composed of two chromatids that are held together by a centromere? So chromosomes are held together by a centromere, so it must be anything before anaphase, because anaphase is where the centromere will split and the chromosomes will be, or the chromatids will be dragged to opposite ends. So the answer is metaphase and prophase. So in telophase, the chromatids are already at different poles, so these out. And A and B, it contains anaphase. So in anaphase, the splitting will occur, so the centromere will be broken, right? And some chemicals used to stop tumor growth work by preventing DNA double helix from uncoiling and separating. During which stage of the cell cycle do they act? Anaphase, interphase, metaphase, prophase. So I'll give you a while to just read this and try to answer this in yourself. So try not to look at the bottom answer, just try to think of this yourself. Okay, so let's explain this question. So what exactly does DNA need to uncoil and separate for? And this is important because DNA won't be able to replicate if it cannot even uncoil and form two template strands. This you'll be able to answer this better if you have learned chapter 6. So chapter 5 and 6 go in hand to hand. So if you're just learning chapter 5 by itself, so this might be a bit difficult for you to understand, but just try to understand this. So DNA needs to uncoil and separate so DNA replication can occur. And DNA replication occurs in which phase? It occurs in the interphase. So this will affect the interphase. So the answer would be B. DNA molecules in eukaryotic cells must be packaged completely, sorry, not completely, compactly, because their combined length is much greater than their diameter of the nucleus. Which structures are required during the process of packaging DNA molecules compactly? A, chromatids, B, chromosomes, C, histone proteins, and D, telomeres. So this is asking about how DNA molecules form compact structures. So think about the structure of chromatid thread and how does it form. I'll give you 5 seconds to think about it as well. Okay, so I hope you got this answer. It was a very straightforward question. So when DNA molecules, they need to wrap around a protein. They're called histone proteins, which forms nucleosomes. And nucleosomes will be linked to each other by linker DNA. So to form compact structures, histone proteins must be involved. So the answer is C. And I think this is the last question that we'll cover today. And okay, so which statements about mitosis are correct? 1, mitosis results in genetically identical cells. 2, mitosis is involved in asexual reproduction. Mitosis occurs immediately after cytokinesis. Mitosis can be used to repair damaged cells. Again, I'll give you 5 seconds to just read it and think about it, and then I'll cover it. Okay, so let's cover this. Option 1 is correct. It produces genetically identical cells because the chromosome number is exactly the same as the parent cell, and the type of chromosome is the same because it's from the same chromosome. It'll replicate, and the same chromosome is being passed. There's no genetic variation like occurs in meiosis. Meiosis will be taught in your A level syllabus, so you don't need to worry about that. Meiosis has genetic variation and mitosis does not. So mitosis is involved in sexual reproduction, yes, because cells are genetically identical. Mitosis occurs immediately after cytokinesis. No, that's wrong. There's an interphase, there's G1, S phase, and then G2, and then the entire mitosis. And then after that is cytokinesis. For mitosis can be used to repair damaged cell? Absolutely not, because as we discussed in the function of mitosis, mitosis is used to repair damaged tissue. It does not replace the cell by itself. It replaces the damaged cells, thus it's repairing the damaged tissue itself. So the answer for this is C, 1 and 2. Only 1 and 2 is correct. So I hope you found this lesson useful. And as usual, if you enjoyed the video, like, comment, subscribe. Do share them with your friends so more people can benefit from the video. If you have suggestions, leave them in the comment section. If you have questions, leave them in the comment section as well. And I'll see you in the next video, chapter 6, which is translation and transcription. And I hope I could make more videos on answering past year paper questions, because I think that would be more useful for you guys. So if you have any more requests, also do leave them in the comment section. So I'll see you in the next video. Happy studying. All the best for your exams. All the best for studying.